Hey everybody, in this video we are going to talk just a little bit about what we can do with SNP profiles, and there's so much we can do, but I think one of the most interesting things is something everyone can do, is we can get our SNP profiles determined by companies like 23andMe or Ancestry.com, and they can give us health information. They can also do ancestry information too, and we don't have time to cover how they do that. But I just want to touch a little bit on the health information. And I think, I have used 23andMe's services, and I think they have like 100 different reports now, maybe more, 100 different health reports. And it's rather interesting, and we are going to take a look at how they, just one of these reports, to see how they can derive health information from our SNP profiles. So we are sort of out of time right here for lecture 21, so I will just kind of go through this as quickly as possible because I don't want to cover all the basic stuff that we did in videos 2101 through 2104, and then not get to talk about the really cool things like this. Well, it's all really cool to me, so I hope you think so too. So in this video, we are going to take a look at 23andMe's lactose intolerance health report for its customers. So now this report is based off of a study called or by Anata et al. Way back in 2002. And so this is essentially what the scientists at 23andMe need to do. They need to go into the literature, research literature, and they need to find studies that looked at single nucleotide variants or single nucleotide polymorphisms and correlated those with a health trait. And then they can come up with a report for its customers. And those reports have to be approved by the FDA before they can be released to their customers. So, well, in this study, these scientists looked at a Finnish family. And in this Finnish family, and I forget how many members are exactly in the family, but it was a rather large family. And there were members of the family that were lactose tolerant and other members that were lactose intolerant. So what does that mean? Well, lactose is found in dairy products, right? So lactose intolerant people can't really digest lactose easily, and they get bloated and feel really uncomfortable after eating too many or too much of a dairy product. But when they're looking at this Finnish family, and what they found was some members were what we call lactase persistent, and others were lactase non-persistent. And what is this? So lactase is an enzyme produced by mammals, and it's a lot like the LAC-Z gene in E. coli. So lactase is an enzyme that degrades lactose or metabolizes lactose into glucose and galactose. So lactase is the enzyme. And lactase persistent people, so these individuals, their intestinal cells continue to produce lactase into adulthood. So cells of the intestine secrete lactase into the intestine, and the lactase can degrade the lactose sugar. Lactase non-persistent cells do not secrete lactase. Now, these are funny words to use to describe these two different phenotypes, right? And the reason we use these is because all infants, like human infants, and I guess before toddlers, what do you call the infant before they're not quite a toddler, like 1-year-old, 2-year-old? I guess infants and other young humans that are surviving on breast milk, well, they need, their cells always produce lactase, right? They need the lactase to digest the lactose in breast milk. But most humans throughout the world, the cells of the intestinal lining stop making lactase. We don't need lactase anymore because, you know what, at two years old or whatever, you stop surviving on breast milk. And so it's inefficient, I guess, and that's why the cells stop producing lactase. However, there are some populations, and I think it's somewhere in Europe, where there was a mutation that allowed some populations to produce lactase, or the cells of the intestine to produce lactase throughout the life, throughout adulthood. And there are theories that, you know, this helped some people, I think, survive famine in the Middle Ages or something, when there were lots of cows around, but and people started to survive on dairy products. I'm not sure if how widely accepted that theory is, but it is a theory that offers an explanation for why this mutation that causes lactase persistence exists in some populations, some European populations. But that's too much information. We're just interested in how 23andMe uses this study to give us our likelihood of being lactose tolerant or lactose intolerant. Okay, so let's get back to this study. Anatta et al., 2002, Finnish family. Some members were lactase persistent, had cells that were lactase persistent. Others were lactase non-persistent. So you can bet the lactase persistent people, these were lactose tolerant. So yep, they had no trouble digesting dairy products. The non-persistent people, lactase non-persistent, were lactose intolerant. Okay, so what these scientists did was they looked at the lactase gene in all of these people here. And what they found, essentially, let me see, how did I want to address this? What they found was in the lactase persistent people and the lactase, or family members, lactase non-persistent, for the lactase gene, which is called LCT, what they found were no differences. No differences in these genes. Wow, okay. But they looked next door to the gene next door to LCT, and let's say next door gene. And they found a difference. They found a difference. And what they found was that if they're in the lactase non-persistent individuals had this genotype that was CC at one position. Okay, so let me see this next door gene. What they discovered essentially was an SNP. Now they call this SNP CT13910, which is an unusual number, but that's what they named it. And technically, it's probably more of an SNV back then, but the definitions of SNVs and SNPs have changed since 2002. But they called it an SNP, and they named it CT13910. And what they figured out, that in the gene next door, the SNP was in the gene next door to the lactase enzyme, and the individuals who were lactase non-persistent were homozygous CC for that SNP. And those who were lactase persistent were CT, so the heterozygous genotype, or TT. Okay, so they found one difference, and they don't know why. I don't think anyone yet knows why this one difference in this gene somehow allows an individual's cells to be lactase persistent, to produce lactase throughout adulthood. I mean, it's not even in LCT, so maybe it's an enhancer. Maybe there's an enhancer that controls expression of LCT in the next door gene. So we don't know. But the lactase non-persistent individuals were CC. And then okay, so that was just for the Finnish family. So what they did in the study was they looked at other Finnish people that were not in the family. Finnish people from Finland. And they looked at people around the world. And what they found was a 100% correlation CC genotypes were lactase non-persistent. They could not digest or their cells did not produce lactase. In CT or TT genotypes for that SNP, they were lactase persistent. Okay, so really cool study from 2002. Now things have changed since then. So that SNP that the authors named CT13910, well, the name of that now is given this official database name, RS4988235. And so if you were to go to the human reference genome and look for this SNP number, well, you'd find it right here. So there's a G and there's the C. So this C right here, this is on the minus strand now. Right, the human reference genome was published after Inata et al. Published their study. Now this is the minus strand, this is the plus strand. So Inata, what they were doing is when they named their SAP, they were genotyping based on the minus strand. So now we genotype on the plus strand. So we have to translate. We have to translate Inata's results. Okay, so how do we translate their results? So if Inata's results suggested CC, let's see, CC, we'll put lactase here, we'll put lactose here. CC was lactase non-persistent, non-persist. That means lactose intolerant. And the CT and TT genotypes, well, they were lactase persistent and lactose tolerant. It's a little confusing. Lactase, lactose, non-persistent, persistent, intolerant, tolerant. I hope this table helps you keep track of everything. So we need to convert now because Inata was referring to the wrong strand, was referring to the minus strand. So now we know the GG genotype, homozygous genotype for their allele. Let's say if this allele was called SNP 13910, it's now officially called RS4988235. You don't need to memorize that for the exam, right? So GG genotypes are non-persistent and for lactase, thus intolerant for lactose. GA and AA genotypes are persistent for lactase and thus tolerant for lactose. So everyone understand how that works here? So Inata was naming the alleles based on the minus strand. So now in 23andMe and Ancestry.com, they're probably going to name the alleles based on the plus strand, and that's actually what they do. So I don't want you to be confused when you see my 23andMe report for lactose intolerance. Like, where did these G's and A's coming from? I thought we were talking about C's and T's. Oh no, now that the alleles genotypes are based on the plus strand. Okay, so my report from 23andMe, essentially what the scientists from 23andMe do is they go to these reports and they find research and they say, okay, well all we need to do is look at our customer's genotype for this SNP, and we can tell whether they're likely to be lactose intolerant, which would be if you're GG, you'd be lactose intolerant, or if you're GA or AA, well, you should be lactose tolerant, according to this study at least, right? So here's my lactose report. It's pretty neat. Let's see. Tom, based on your genetics, you are not likely to be lactose intolerant. Okay, so that's a double negative. It's hard to understand, but so you are, we could convert that. You are likely to be lactose tolerant. Okay, so this is just their report for, people who don't care about, how the study was done, how they determined that. And you can click a link, I think, somewhere on there. I didn't have it there, but you can click a link to get more information. So the biological explanation of the results, and here they have their references, and one of these references is actually the Anata study. So essentially the marker tested. So they have the original name of the allele, they have the new name, RS4988235, and this is what you can look up on the Human Reference Genome Database. And look, I have 2 A's. So an A from one parent and an A from the other parent. I don't know why they joined these like that. Those are supposed to be two different chromosomes. They have them linked together like that. It's kind of weird. I might have to call them and have them fix that. So, okay, so I have 2 A's here, and thus they're saying that, okay, let's go back to I have 2 A's, so I'm likely to be lactose tolerant. Okay, pretty neat, right? So we can, I haven't looked at other reports yet, but we can probably guess that every health report is going to be based on some study like this. And we don't know why. We don't know why this genotype leads to lactase persistence and thus lactose tolerance. But there's a study which found this study found a perfect correlation between these two, and so it seems a pretty good likelihood that, yeah, I should be lactose tolerant. And I am, as far as I can tell, I am lactose tolerant. My wife got the opposite result. She is this genotype, which suggests she is lactose intolerant, and she seems to be lactose intolerant. So yeah, so far my family, the 23andMe predictions seem to be accurate, and they're based on this study, and it's kind of neat. So okay, so that was a little longer than I wanted it to take, but I thought it was rather interesting. I hope you think so too. We're going to do one more video on this lecture because we need to talk about genome-wide association studies, which can use SNP profiles, SNP genotypes. Those are the same thing. I can't decide which one I want to use. To learn about the genetic basis of complex diseases, to learn about the genetic basis of diseases that we pretty much had no ability to learn about in the past. So, okay, see you in the next video for the very last video of this lecture.